There are certain things that I want you to either tattoo on your forehead or write on your bathroom mirror in lipstick, or, just make a sign and tape it to the wall. And this is one of them. This is a really important concept. When we looked at skeletal muscle, we saw that individual muscle cells were all or none. In other words, if you had a muscle cell reach threshold, it would contract to its full extent or not at all. If it didn't reach threshold, it wouldn't contract. The contractility in cardiac muscle cells is graded. And what makes it graded? In other words, it can contract a little, it can contract a moderate amount, or it can contract a lot. And the key to this, the force of contraction is proportionate to, this is an alpha sign, is proportionate to the intracellular fluid concentration of calcium ions. The force of contraction is proportionate to the concentration of intracellular calcium. So how about a little thinking question here then? If that calcium pump that removes calcium from the cytosol and puts it back into the sarcoplasmic reticulum, if that's interfered with, or what is more common is to interfere with that secondary active transport mechanism that gets calcium out of the cell and back into the extracellular fluid, if that's interfered with, we will see a buildup of intracellular calcium. And when that happens, we will see an increase in the force of contraction. I'll write it this way. If we increase intracellular fluid calcium, then the result will be a stronger force of contraction. We're going to see that in our cardiac lab. When I talked about the ECG in the past, the EKG, I pointed out that the waves trigger electrical changes in the membranes of these cells. The segments or intervals are times between the waves. It may include one wave if it's an interval, and it is during those times between the waves where we see a mechanical action. So the term depolarization and repolarization is electrical, and that will trigger a mechanical event. Here we see that the atria contract during the PQ or PR segment. Again, here is the start of an electrical event, the Q wave, the R wave, the S wave for our QRS complex. When it is completed, we have the ST segments, and that results in a mechanical action. The ventricles contract. And then again, an electrical response, repolarization, that is going to result in the mechanical response of relaxation. So please keep the concept of depolarization and contraction separate from one another. Depolarization is electrical, contraction is mechanical. The electrical events must precede the mechanical events. So these mechanical events, let's get into that. Well, we use two terms to help describe contraction and relaxation. Contraction is referred to as systole, not systolic, systole contraction, where relaxation is referred to as diastole. So we will start using those terms now. Here in this cycle, we see, if we are going to start, diastole. All four chambers are relaxed. Blood is flowing passively into the atrium, which is then flowing passively into the ventricles. Look at how long ventricular diastole lasts, and look at how long atrial diastole lasts, and how long of a period of time the heart is in relaxation or diastole. So during atrial systole, this is when the atrial are going to contract, and it will push a fairly small amount of blood into the ventricles. Our next step is isovolumic, which means same volume. We're not changing the volume. Ventricular contraction or systole. So the ventricles are contracting enough to close the AV valves, but not strongly enough to open the semilunar valves. So although pressure is increasing in the ventricles, the volume of blood stays the same because nothing is open, no door is open here. Isovolumic ventricular contraction. As the ventricles continue to contract, the pressure continues to increase, and eventually the pressure in the ventricle is greater than the pressure in the major arteries leaving the heart. So the semilunar valves open, and you have ventricular ejection. Blood is ejected out of the ventricles and into the arteries. Our AV valves here are still closed. As the ventricles undergo repolarization, you are going to have ventricular relaxation start, and so we're starting to see a decrease in ventricular pressure, but not low enough to open any of the valves. So all four of those are still closed, but as the ventricles continue to relax, the AV valves open, and we are back to diastole. Let me make one more pertinent point. When we look at this, we realize, especially regarding the ventricle is in systole about 1/3 of the time, and in diastole 2/3 of the time. That is key, and we'll see its influence when we talk about blood pressure. Sorry about the blurriness of this image, but I think you can pick things out. This image I cut and pasted from another image out of what is referred to as the Wiggers diagram. We will look at that in more detail in a minute. But I wanted you to, again, visualize and understand the relationship, the correlation between the EKG and the cardiac cycle. So again, here's your ECG or EKG. P wave triggers atrial systole, that is represented by the atria contracting, etc. We'll look at this in more detail in class, but I certainly would get a good start on it before coming to class. Your textbook has this really nice illustration that shows you how changes in pressure over time are reflected also in what we call the Wiggers diagram. And the changes in pressure are going to change the volume in the chambers, it's also going to trigger changes in the heart sounds. So take a look at this illustration. And again, we will put all of this together, what you saw in the previous slide, what you see in this slide, and I'm going to show you more pictures, but we'll definitely look at it in class. So this is the big picture, what is often referred to as the Wiggers diagram, that lines everything up. Here we have your EKG or ECG. Next we have changes in pressure, in particular the left ventricle. That is our default chamber, if you will, because it's the one that pumps blood out to the organs, in particular the brain. And if the brain's not happy, nobody's happy. Underneath the pressure graph are heart sounds, and then below that are changes in volume in the left ventricle. And then finally at the bottom we have the cardiac cycle. So again, we'll see this in class. Oftentimes when I post something like this, students look at it and freak out. But if we go through it step by step in class and you understand it, you will have probably about 75% of cardiac physiology. Here is your textbook's version of the Wiggers diagram. Again, we'll look at it in class together.